And next, we'll just move on very quickly. I would like to introduce Bjoern to the virtual stage to present the flip side to the database, which is the analysis resource. Thanks very much, Bjoern.
Thank you, Nina. So I should be sharing my screen now. Yeah. Okay, so like Nina was saying, I'm going to be presenting the analysis resource overview. So key here is this is an overview. We have day 2 tomorrow, which is completely devoted to the analysis resource. So today, in these 20 minutes that I have, I'll just give a very quick summary kind of what's provided in the IEDB in terms of the analysis resource. So why is this not going away? Sorry, 1 second. Hide these Zoom controls. Okay. Briefly, the IEDB is separated into 3 main groups of tools, those that are related to T-cell epitope prediction broadly, to B-cell epitope prediction broadly, and analysis tools. And some examples for each of these categories is listed here. There's actually more for each available. And again, as I said, I'm going to give some brief examples here of what these tools are and introduce some broad context that are going to come up across the workshop presentations, also to kind of familiarize yourself with the vocabulary if this is new. If you're familiar, if you're a constant tool developer, much of this is going to be not news to you. So the prediction tools that we have for both T-cell and epitope, B-cell epitope predictions are based on machine learning. And what does this mean? That we have experimental derived data sets that are used to train a machine learning method. So they might look like this, that you have training data where you have individual peptide sequences that have a measured binding affinity to a given MHC molecule. But you could also have 3D crystal structures of, say, antibodies binding to proteins. You can have T-cell assays of having cytokine production after a vaccination for certain peptides from the vaccine. And that data, obviously, this experimental data that is used to train these methods comes from the curation process that Alex has introduced you for the IEDB database. There's various different prediction methods, prominently neural network, preferences of what kind of method to use. And the idea is that these are looking at, oh my God, my internet connection is unstable. Nina, am I clearly audible?
You are okay now. We just had a momentary pause from you, Bjoern, but you're good.
Okay. Well, I'm hoping this is gonna stick. I mean, worst case, I can turn my video off while I'm presenting. But okay, let's see how this goes. I had no trouble for the last 24 hours anyway. So then this training data is used to train a prediction method or model based on these machine learning techniques. And then this can be used to have new input data, so new experimental data where you don't know the experimental outcome, and extrapolate based on the patterns identified from the training data, what the likely prediction results would be. So that broadly is what we mean by prediction models. So to get to the IEDB analysis tool resource, you can go directly at tools.iedb.org, or you can go to the main website, iedb.org, and then in there, you can either click on this analysis resource tab that takes you to the main tool homepage, or this one, or you take the specific tools of interest that you care about. And this is all on the homepage to like signal to people what kind of tools we have available on the site. So once you're at the analysis tool homepage, you have different categories available that are accessible through these tabs. These are the 3 that I just mentioned. And then there's also different ways of accessing the tools. There's a tools API / RESTful interface that can be used to programmatically run these tools and not on the website. Then there's the ability to download standalone tools that you could run on your local machine. Then there's the data sets that are used for training and testing of the tools. And then there's, as everywhere on the site, access to help tutorials, a solution center where you can ask questions, and direct contact to the IEDB. That's actually the same thing. The contact goes into the solution center where you can ask questions. So looking at the T-cell epitope prediction tools, this is kind of when you click on that, you're getting the breakdown of the tools that are available. And then, for example, for MHC binding predictions, which I've been introducing before, this is then how the tool looks like, the web page looks like when you access it. So essentially here you have a form in which you're defining the problem that you're asking the tool to examine. So this starts with entering the sequence. In this case, might be a sequence of a protein that you want to scan. And then you have to select the prediction method that you want to use. As I said, we have multiple methods available. We have this concept of an IEDB recommended method. Because there's many methods available, and it is hard to compare. So a lot of effort actually goes into, on our side, to benchmark the different prediction methods that are available and compare their performance on test data sets that they hadn't seen before, which we then publish, and based on that choose. Here in this case, this is the last large benchmark we ran for MHC binding predictions, which showed that that was run in September 2020, that in that case, the EL 4.1 version of that MHC PAN performed the best. So apart from the methods here for MHC binding, you have to identify the allele you want to make predictions for. Then you can also, if it's a very large set, you can let them send to you by email. If not, you're going to get a result section like this where the sequence is echoed. You have the individual peptides that are found in the sequence ordered by the prediction score. And in this case, it identifies that the high score is a good binder for the specific method output. So the same thing is available for MHC class 2 predictions, very similar setup. And for both MHC class 1 and class 2 predictions and the predictions around them, we have identified the need to really guide users through the process, because when you run this, you typically end up with like this large table of peptides, and people have been saying, well, what do I do then? Which one should I choose? So here, this TEPI tool actually uses the same predictions, but has a number of helper steps around that go from 1 through 6, which then essentially define the problem more clearly and ask you some questions about like what do you want to see, and then give you a condensed result like this that are saying, okay, this is actually the peptide you should be looking for. And you have different forms of the results that are essentially doing like additional processing steps to get to a more actionable result than a very large table. So for B-cell tools, again, the setup is very similar. You get here by hitting this tab on the tools.iedb.org page. And the biggest difference for B-cell epitope predictions is obviously, so we say B-cell epitope predictions and antibody epitope predictions interchangeably. In all these cases, you have an antibody binding to antibodies obviously produced by B cells and are actually found as surface receptors on B cells. But then ultimately, it's the antibody CDR regions that are binding to an epitope of interest. And that is a structural process, but you can still ask, okay, if I give the protein sequence in, a protein sequence of interest, which are regions that are likely going to be harboring B-cell epitopes. So this is what we consider a linear epitope prediction. And you have a number of methods, again, to choose from. When you run this, you get a result here back that is visualized, where you have a score from low to high. High here, this is the thresholds considered to be significantly high enough to be like the epitope containing region. And then these are not just graphically displayed, but also as a table of these regions that you can then examine and look through. So and this you can modify how you want to specifically summarize this, but by default, these are set to what was used in the publications. So, but as I said, ultimately, this is a structural problem. We write a sequence of a protein as a linear stretch of letters, but those amino acids form a structural conformation when they fold to a protein. So these are the structure-based B-cell epitope predictions, and one of them is Discotope, where you then enter not a sequence, but a PDB file. And PDB stands for Protein Database, Protein Data Bank. I got this wrong. Which is like the reference database for crystallographers came up with this. Essentially, it's a reference format how to describe 3D structures of proteins, and they have these PDB codes, but you can also use your own file. And now there's prediction models like AlphaFold, which we learned about, that you can use instead as your input for what do you think the 3D structure is. And then the Discotope method, again, scans the sequence, but actually does it in a 3D format. You can visualize the results like this here, but you can also have, so that's the table view, but you can also look at the 3D view and ask, where is this on the structure, where then you have the regions highlighted that are actually likely going to be binding regions of antibodies. So I mentioned this up front. This is what I showed you right now is the web page that you can directly look at in your browser and run things in your browser. But we have standalone versions of these tools available, I think for the like 80–90% of them. And you can then use these on your local machine with the advantage that you don't need to send large amounts of data over the internet. If you have very large data sets, this is also going to be a way to run them much faster than running them on the server. This is free to use for a nonprofit in academia. And there's, if you're working in an industry, there's a nominal fee that we're asking for when you use these tools. This is how these tools then look like in practice. You have a script here, which you give as an input then some parameters like the what you would not otherwise enter in the web form, and you give it a sequence file as an input, and then it gives an output like this that you can incorporate into your own whatever, save it as a file, whatever you want to do with it. And this is how the results look like, essentially the same as you have on the web page, but as a file. Then we have an API version of the tools. So this is where you send a prediction request to the tool server at LJI through a script. So there's no need to install tools on your local machine. These are free. This is freely available to all users. And the idea here is also that as tools change over time, you don't need to go and reinstall and troubleshoot the local installation, but you can benefit from updates that are happening on our server, and they essentially then just become available in terms of like say there's going to be new strings they can use to select new versions of tools. Yeah, this is how the API version can be used. So you say you have an Excel file here, that a CSV file of your peptides of interest and certain alleles of interest that come out of the research that you're doing, and you want to predict the binding affinity of each of these peptides to each of these alleles. You can write scripts like this where ultimately the command to get the prediction back is that you just have like these curl calls, which is essentially going straight to the API interface, and you're passing on some commands and getting, in this case, the consensus percentile back, and then you just read the file, you go through the peptides, and then you have your output file that is served as predictions results.txt, and this is then what you have here. And again, like we're going to talk about this more tomorrow. So let me summarize the different versions that we have available. There's the web page, which I started off with, which you can get at tools.iedb.org. I mean, this is the main one. And then I showed you how to get to the individual tools, where you use your browser to submit data. You run predictions on the IEDB tool server and get your results back. This is anything that can run a browser can run these tools. Obviously, you need internet. And for very large data sets, our servers are capable, but then you're going to run into issues with like data upload and download and stuff, depending on your internet connection. The advantage is these are automatically updated. Now, if you download a standalone, then you can use your command line interface, which requires a little bit of programming experience or computer experience. You download them from the IEDB website. You can run them on your local machine. These are only going to work on Linux. That's what our servers run. And you can use virtual machines to run Linux code on other operating systems. But yeah, this is an additional, obviously, potential hassle. You don't need internet. That's an advantage. You can run as large as a data set as you want, as a computer can handle. You will need to update this every time there's a new release. And again, like these are for industry, there would be a fee involved. You can also run the API, which you can do API calls on the command line interface or integrated into your code. You have these are run on the IEDB server. So you essentially send a prediction request to the server and get your results back. You do need internet. You can make custom scripts and for your custom results and process them, which can be really neat and benefit from the automatic update on the web server. And this is free to all. So that's actually my last slide to remind you. And actually, I kind of said this, but not really. So these are the prediction tools. And very often when people approach us for, I want to predict X, we always ask, have you looked if somebody has done your experiments before? And you don't need to predict, but you can actually look up the results. That should always be your first step, to go to the IEDB database and ask, can I just look up rather than predict? So the Epitope prediction tools that I showed you extrapolate from existing data to identify new candidates, to extrapolate experimental results to things where we don't have experiments. This is done by machine learning approaches that essentially identify pattern in your training data and then predict new data. We're using our C-curves and AUC values as our preferred performance metric for these methods. And again, we're going to talk about that more tomorrow. I do want to point out that by these predictions, I think they're working well, but at the same time, it is not that you would run a bunch of predictions and say, now I have like a set of peptides that I think are a good vaccine or so. You should always experimentally test these things. These are meant for candidate identification, not for like, you can't take them as ground truth. The analysis tools, which actually Alex is going to talk about later today, they are really meant for examining sets of epitopes and gain new knowledge about them. So they're not prediction tools, but they're taking existing epitope data and are used to do things like finding clusters of related sequences in a list of peptides or so that come up over and over as we are when we deal with epitope data sets. So that's why they were implemented in the IEDB, and there's a broad array of applications for them. That is my last slide. And I will click to exit and stop sharing.
Excellent. Yeah. Thank you so much, Bjoern. We are running a couple of minutes ahead of schedule, so I'm just going to quickly add a few reminders before we move into the Q&A. So for those of you who have joined recently and maybe missed the introduction, we look forward to receiving your questions in the Q&A function at the bottom of your Zoom webinar. Please don't post your questions in the chat. The Q&A is just much easier for us to manage. So thank you very much for that. We've already had a few questions come through, so I'll give you a few more minutes to submit those. But the first one I would like to clarify, I did answer it by typing, but in regards to the slides for the user workshop, you will receive those slides in your inbox approximately a week after the conclusion of the workshop. So we will be sure to send you a post-event email, which will contain a link to the presentations, a link to the YouTube videos, which we will be uploading from the recording of the 3-day event. So you'll have all of those in your inbox about 7 days after the conclusion of the event, with our post-event survey as well. So just a few reminders there. But now I would like to call Alex back to the stage and Bjoern to tackle the questions in the Q&A. So Alex, would you like to go first?
Yes, so this was a question from Alessandro Pina, and the question was about the curation process. The data is curated from the present time to 2012, red line, is it correct? No, it's not. I apologize, I was not clear. So in 2012 was when we caught up. So essentially what we did when we started the IEDB, we started curating new records that were coming in, and at the same time go back in time and curating progressively older and older references until we reached the quote unquote end of time, in the sense we curated the oldest, the 1970s antibody references in the IEDB. And at that point, that happened in 2012. So from 2012 on, we have been only curating new references that are published in the scientific literature, and to a certain extent, every once in a while, also PubMed enters new references that they found or whatever. But yeah, that's the answer to a question.
Thanks, Alex. And Alex, I accidentally pressed I would like to answer live to the next curation question, and I can't undo it. So I hope you
okay, I can answer that as well.
Okay.
There is a question from Ghazal Azari, and the question is, can you briefly explain how the negative data is generated? I guess we do not have them from the experiment, and how are they generated, right? So the negative data, we don't generate any data, negative or positive. It may be that a paper will have negative data. And for example, two common instances of negative data is someone says, had generated overlapping peptides spanning an entire protein and tested them in mouse that were immunized with that protein. And so there's going to be data, and that person might have found in the experiment that peptide number 3, 5, and 12 were positive, but all the other ones were negative. And so we curate that, and that record is entered as such. Another instance might be that, say, a person has been testing responses from individuals infected with virus XYZ using a particular peptide, and might have found that that particular peptide elicited CTL, the T cells that were making interferon gamma, but not IL-4, or in an allergy setting, for example, we say that allergic people were making IL-4, but not interferon gamma. That would be curated, the positive and the negative assay as well. Thank you for the question. It's a good question to clarify.
Okay, I'm going to answer a few tool-related questions that came up. So one of the questions was that the AUC curves that I was showing only include summarized data for all MHC alleles included in that set. I will go into more detail about that tomorrow, where we're going to show like where the breakdown can be found. And those curves actually they were mice. So I can go into more details tomorrow. That might be, unfortunately, an answer to several of the questions that I have. Then the next one here, sorry, that was Mauricio Calvo de Cal that asked that question. The next one is from Akbar, who asks, does a high EL score mean it not only has high probability of being eluted, but also that it has high binding affinity? Can a poor peptide binder have high EL score? So that shows some detailed knowledge. So the EL tool is trained on MHC ligand elution data in addition to MHC binding data. And essentially, MHC ligand elution data has become a much larger set of data than pure binding affinity measurements. And what has been happening over the past few years is that you have tens of thousands or hundreds of thousands of data points from ligand elution experiments. And ultimately, for a peptide to be found in the ligand elution experiment, where you essentially sample the surface-bound peptides on a cell, these peptides are always high affinity binders. So whenever we have run experiments to really go into detail and looking for a peptide that is like seems to be bound on a cell based on a mass spec analysis of peptides that had a low binding, we typically could identify that this is not really a binder. And definitely the algorithm that generalizes over tens of thousands of points learns this, and there are essentially no peptides with a poor binding score and a high EL score. And that's why actually that method works well for binding predictions as well. Alex?
Yes, so I'm going to answer two questions. One was, can you elaborate? Let me find it again. Can you explain from Tekiba Sisai, can you explain the minimum length of an epitope? Right, so here we had to be defining an operational threshold. And this goes back to how epitopes are defined and reported in the literature. So as I was mentioning, we set a minimal size of 50, arbitrarily of 50 amino acid or 5,000 Dalton, because we figure that everything above that is really not defining an epitope, but is defining a domain perhaps, or a broad structure. Now, the important thing, and this question is great because it gives me a chance to elaborate a bit on that, is that clearly, when you say there's data that say a particular antibody reacted with those 50 amino acids, doesn't necessarily mean that every one of the 50 residues in that fragment is part of the epitope. In fact, it's quite likely that if the data would be defined at the higher granularity level, you would define what are the smaller subset of epitopes. So in that we say that those 50 amino acids, it's a fragment that contains the epitope, but we don't know exactly which one are the particular residues that make up the epitopes. There are cases where that is absolutely known. Say, for example, when an epitope, the minimal epitope is defined by truncation analysis for a CD8, or an eluted peptide is defined, which by definition that is the peptide, or there is an X-ray crystal structure that shows what are the residues that make contact with the antibody or the B-cell receptor. Then there is a third category where, say, someone has studied, again, an antibody and defined a set of mutations that are key, and the mutants that have been selected to lose reactivity with antibodies tend to cluster around those particular residues. Now, those are residues that are important for the epitope, but say that the study defined 3 residues that are important for a particular antibody. That doesn't mean that those are the only residues in the epitope. So that's another situation where those are a partial definition of the epitopes. We know that those residues are important for the epitopes, but in all likelihood, there is other residues. I also am going to take another question from Mauricio Caldevalle that asks, how long does it take for an epitope reported in a paper to be included in the IEDB database? And the answer here is a statistical answer, in the sense, actually, we monitor that very closely, and we actually report that data to NIAID on a constant basis. And we set for ourselves the goal to achieve 80% completion of a curation of new references that appeared in the PubMed query in 12 weeks from when the publication has arrived. And in fact, I'm looking at the data right now, and we're closer to 10 weeks. And also we set the date for 95% completion at 18 weeks after publication. To get 100% completion is difficult, because sometimes, for example, it happens that a paper is published and they say, oh, use the residues 18 to 27 of hepatitis, and we have to contact the author and say, well, which hepatitis? I mean, what are you talking about? Which is the particular gene bank accession number that you use to make that peptide? And so obviously there we are dependent on sometimes finding out additional information that doesn't entirely depend on us.
Thanks, Alex. So next question is from Sumyadeep Chakraborty. Can you elaborate more on this predicted score for B-cell epitope prediction? How is rank values different from the predicted score? Which to choose? So typically a method that is being developed, pretty much every single one that we have will have its own kind of generic idiosyncratic output. And we often, or for nearly every example, we are providing rank-based predictions instead that are using essentially a large reference data set that is being predicted, and then the scores are taken for this reference data set and we convert them to ranks. The advantage of the ranks is that then you have a score that you can predict across multiple different prediction methods that might have their very own scale, but then you can combine them overall into consensus better, so it makes it easier to compare. If you're just doing one method at a time, typically rank or direct score output doesn't make any difference at all. So that one, then the next one was from Remy Crusoe, and I totally apologize for butchering all the names here. Does the T-cell epitope database provide TCR data? For example, CDF3 sequences known to bind an epitope on the specific HLA? Yes, it does. And I believe that's going to be demoed today. So there is like a T-cell receptor tab, or there's a receptor tab that has, if it is available experimentally, an annotation of the sequences for TCRs or antibodies. And I know we're running a little bit late, but I think we can do this, Alex. Go for it.
So Gia G asked, can we identify web applications that are related to a particular epitope in the search results? As a general rule, yes. So whenever you have a search result, you have also tabs or things that you can click on related to the references. And then when you click that, you will have the list of the references that are related to that particular query.
Okay, so then I can take the next one from an anonymous attendee. Do all antigens have epitopes, and are all epitopes part of antigens? So all the antigens listed in the IEDB are listed there because there's at least one epitope in which they are found. That's why they're essentially being added. The other question, are all epitopes part of antigens? So that is also semi-philosophical. If somebody says, well, if you're testing a peptide that is completely artificial, and that might be that you just are interested in generating random peptides and ask how often they're recognized, that would not have an antigen as its source protein. But the vast majority of epitopes that we have an antigen, and we always try to identify it for any kind of natural peptide. Alex? Or are you done?
I'm done. I think.
Okay, so I always do the done thing. I see them all still there. Then I'll do the last one. From Daniel Hakim, I have run NetImageD Pan 4.0 to come up with a potential epitope of interest for a specific HLA. What do I do next? How do I evaluate whether or not this is a epitope is real? So again, like the first thing would be, I mean, is it in the IEDB? But then it really depends on what kind of question that you're having. Yeah, are you asking, is this a specific peptide that you think is recognized in people who have been infected with influenza? Well, then the best way of testing it would be to get blood from these people and test that in say in an ELISpot assay or so. Is the peptide being recognized? You have to make it, you have to do the experimental work. There's other things, it really depends. I mean, if you're looking for a mouse peptide, obviously you should do a mouse experiment. If you're all these things. And I think that was the last question.
There is, Bjorn, there is one that was in the chat from Tom Jang that asked, are there suitable tools for TCR peptide MHC prediction? I think that we're going to talk about it next tomorrow, right?
Yes, Yes. So I think we can stop here, Nina.